you have any question?
So if there are no more questions to be asked, let's move to the next presentation from Duran Yu, Senes S. The title of the presentation is Biochemical Effects of Live Fish Transports, a Comprehensive Review.
Thank you, Dr. Chamanti. I have recorded my presentation. If it's possible, can we play it, or I have to present here? Okay, thank you. Greetings and good day. First of all, I'd like to thanks to conference organizers and secretary for their outstanding efforts in making this event possible. Welcome to my presentation. Today I'll be delivering a presentation with the title Biochemical Effect of Live Fish Transports. This presentation will focus on the biochemical effects of transporting live fish. Process of live fish transportation encompasses a complex interplay of factors that extend far beyond the just physical relocation of fish. In this review, we focus on important factors to consider to increase the success and animal welfare of live fish transfers. Live fish transfers are critical in aquaculture and can be made more efficient with careful planning and good practices. Open system transport vessels with oxygen connection are used in commercial fish transfer operation. As you see, there is fish loading process from cage to transfer tank, but there is air exposure here. For example, as you see, fish loading to cage nets and taking from cage to transfer tanks, and then exposure air here. The pivotal moment during this transfer processes is the exposure of fish to air during loading, recognized as the most stressful event in the transportation of salmonids. What is stress or stress factor? The physical response of the organism as a result of the intervention of external or internal factor is called stress, and the stressor is called a stress factor or stressor. To reduce the effects of stress and improve animal welfare, it's recommended to minimize the duration of exposure to air and avoid exposing fish to supersaturated and suboptimal dissolved oxygen levels. Transporting fish from farm to outlet, transferring to a new cage, scaling exposure to air, loading, transportation, and unloading are all stressful for fish. In a study conducted by Iverson et al. In 2005, it was shown that fish loading procedures are more stressful than the transportation itself. The process of loading fish into tanks before their transfer represents a physically demanding task for both aquaculturists and the fish themselves. The environmental factors affecting trout are routine farm practices, environmental pollution, and seasonal factors, all of which can be considered as stressor for fishes. Farm practices cause changes in parameters such as oxidative index, glucose, and cortisol as a result of the excitement they cause in fish. Fish weight and water temperature are important factors underlying the oxygen consumption of fish in relation to oxygen metabolism during transportation. The increase in water temperature in transport tanks increases the need for oxygen. In terms of fish transportation, for every 0.5 degrees increase in temperature, the stocking density in the tank should be reduced by about 5.6%. Conversely, for every 0.5 decreases in temperature, the load can be increased by about 5.6%. Oxygen. The amount of oxygen a fish consumes also depends on the amount of oxygen available. At high levels, the fish will consume at a constant rate. When water oxygen levels are low, fish consume a lower amount of oxygen than when oxygen levels are high, despite the degree of activity. When dissolved oxygen in the water is lower than needed, this is known as hypoxia. Water without oxygen is called anoxic and it's lethal to fish. Hyperoxia, on the other hand, provides too much oxygen, and while this doesn't threaten to limit aerobic ATP production as hypoxia does, hyperoxia can affect a wide range of physiological and biochemical function in fishes. Carbon dioxide, pH, and ammonia, high carbon dioxide concentrations are harmful to fish and are limiting factors for transfer. Carbon dioxide, a product of fish and bacterial respiration, acidifies the transport water. Although this reduces the percentage of non-ionized ammonia in the water, it also reduces the oxygen carrying capacity of fish blood. Although oxygen levels are apparently sufficient, fish can die if carbon dioxide levels are high. Cortisol. An increase in plasma cortisol levels is expected to be common in all stressors. Studies have shown that plasma cortisol levels in fish, as in other vertebrates, are recognized as the main determinant of stress. In general, glucose is required by all body cells as a continuous energy source and must be maintained at adequate levels in plasma. Perchin and Konyaloğlu, 2008, reported in a study that glucose in blood serum may be one of the best indicators of stress in fish. Depending on transfer condition, fish show extreme effort and a large amount of muscle activity. When muscles are actively used, there is not enough blood to meet their needs. An alternative system comes into use where energy is provided in the absence of normal amounts of oxygen. Lactic acid accumulates in the muscles and blood and causes the pH of the blood to drop. Oxygen utilization is reduced by the low pH of blood. After a few minutes of strenuous muscle activity, lactic acid accumulation may not decrease for 24 hours. In many studies, it has been reported that lipid peroxidation formation in blood and tissues starts with the increase in serum MDA malondialdehyde level due to stress in fish after transplantation. When the stress caused by live fish procedures in fishes is evaluated biochemically, it causes an increase in plasma cortisol and glucose levels, as markers, and a decrease in antioxidant capacity due to lipid peroxidation. Stress can be minimized by following good handling procedures. Proper design and operation of transport tanks and oxygenation system can reduce many fish stresses. Proper water quality is important to maintain fish health and reduce stress during transportation and adaptation to the water which they will be transported. Thank you for listening to me.
Good day, everyone. In behalf of my team, may I humbly present our paper entitled Distribution and Diversity of Freshwater Fish Species in.
Thank you, Dr. Utku. So let's move to the Q&A session. If anyone have questions, feel free to ask him, or you can drop in the Q&A box.
Can I ask?
Sure.
Yes, please
Hello.
go.
Hello, Dr. Duran. Is it possible to determine the duration of stress by the level of cortisol and other substances in blood of fish? For how long time this stress?
Okay, firstly, thank you very much for your question, Olga. Cortisol is second significance for stress. Firstly, glucose, and then cortisol. There is some research about the cortisol levels in time. For example, one research says 2 hours later, cortisol level is the peak, and then the cortisol level 4 hours later, and cortisol level was the same with the normal situation. That means for fishes, cortisol level after the glucose increases and increasing, but almost 4 hours later, it must be get down, decreasing normal set level. But we have to get some more research about the cortisol levels, I think, because, for example, my thesis about transfer live fish in trout, and we got fishes from dam lake to sea with transport. We have seen that cortisol level was higher in control group, I mean in dam lake, and then decreasing to transportation time, and then after transportation it was increasing again. I think we have to get new research about the cortisol levels. That's the good question. That's the critical point, I think so. But we have to be careful about the blood sampling, because in when we are drawing blood, we have getting a new stress for fishes. If we healthily check cortisol level, we have to get so careful, and we have to take them different fishes groups with different hours, I think. Good question. Thank you very much, Dr. Olga.
Yes, thank you very much. And more questions. In my country, live fish transportation for boat, in special boat, this water in boat.
Yeah.
Yes. The maybe perhaps stress will be reduced in the fish transported in the water.
Yes, I think so.
Yes.
And this is the different type of.
But in my country, cold water.
Cold water.
Yes.
Which species? Trout or the different fish? Trout. Yeah, rainbow trout.
Yes.
For example, I have to show something for the transportation. I'm so sorry.
What? Okay. Okay. Sure.
As you see here, we are transporting fish to truck, and then we have sent to the seaside in boat. For example, here. So sorry. Yes,
Sorry.
I think you have seen, and here, I mean, this guy with red shirt, there was transporting with a boat. I mean, big boat, almost ship, with oxygen supplying again. And I think this is not. I think trucks transportation is more stressful for the fish, because
Yes,
here
yes.
water taken from the sea and then going to tanks, and this is fresh sea, fresh water, and
Mhm.
metabolic. I mean, ammonia or carbon dioxide is going to the sea side and taking fresh water from sea to tank, and I think this is the best way for the fishes transportation.
Yes. Thank you very much.
You are welcome. You are welcome. Thank you. And I have to say something to the other participants. I'm so sorry about this situation because there was so a lot here, and I'm in hospital now for my son urgent heart surgery, and we are waiting him on the garden of the hospital. And here I have to do this presentation here, and there is so lot. I'm so sorry about this situation. Thank you very much for listening to me.
Health for your son.
My son,
Dr.
yeah, my son getting a heart surgery here now, and
Dr.
he's in operation
Dr.
now.
Doran,
Thank you very much.
Dr. Doran, I have a question.
Yes, thank you. I'm listening.
I have a question. You have very nicely reviewed the previous methods for the transportation of fish, right?
Yes, thank you. Thank you.
Do you have, or you studied, are you any extracted some sort of standard for transportation of fish?
Mhm.
That water quality parameter should be like this for the safe transportation in fresh water, in sea water, or at this temperature, at this temperature, like this. You have any something like this?
Yes, thank you, doctor. We were tried this for the trout fish, and from dam lake to seaside. I mean, releasing to sea, and we transport with them with truck, and then after the boat and ships. And there was the other type of transportation live fish. For example, tuna. I mean, bluefin tuna. They are transporting in the cage in sea from ocean to the other offshore side to sea for with cage in sea, and this is different biochemical. I think biochemical change this transportation, but we were checked that MDA, glutathione peroxidase, and total oxidant and total antioxidants, and also AST, ALT, and the other biochemical parameters. And we have seen that all of them, the different changing. If we want to check transportation effect, we have to get a new group with them, because for example, one fish, for example, tuna fish, bluefin tuna, it's there was no changing about AST, ALT, or the other biochemical parameters, but MDA and also glutathione peroxidase is different. It's different. It's changing because of they are in the sea when they transportation, but in trout all of parameters changing about, for example, lactic acid and the pH, blood pH, and AST, ALT, because the liver damage in transportation because of the metabolic waste. And if we want to learn something about the transportation biochemical effects, we have to check with different groups.
No, every group has different conditions for transportation, right?
Yes, I think so. I think so. Because of the species differences and the transportation condition differences and the metabolic mass differences and the stock density differences about the all environmental change is different for all transportation. If we want to be sure about the image of biochemical, we have to check the other groups with the other groups or control groups.
But do you recommend any ranges of oxygen, pH, ammonia for safe transportation at
Yes.
particular temperature?
Two types transportation here. One of them the closed system. Closed system when just consumption oxygen from outside the tanks, and this one there was a lot of oxygen. I mean hyperoxygen here in tank, and carbon dioxide is not so much, and pH level is going normal. But the other types, I mean open system, there is no oxygen consumption in the tanks. This is for the other fishes. I mean, for example, small fishes, and there is carbon dioxide level increasing in water, and pH level is going down, and blood acidosis is getting in this transportation.
Okay, thank you, Dr. Doran. Thank you very much.
Thank you very much, Doctor.
Thank you, Dr. Duran. And we wish a very healthy recovery for your son as well. So if there are no more questions, let's move to the next presentation. The presentation is presented by Gepilano KG, Dizon J, Gavina DJ, Lagas JJ, and Tesoro BV. The title of the presentation is Distribution and Diversity of Freshwater Fish Species in Selected Areas of Baikal River.
Hey everyone, in behalf of my team, may I humbly present our paper entitled Distribution and Diversity of Freshwater Fish Species in Selected Areas of the Bicol River. Rivers are one of the common sources of livelihood and a vital source of food. River health condition is the basis for conservation and management schemes and can be integrated into regional and national environmental policies. Rivers provide both economic and ecologic benefits. Little River is the area of the study. It provides a wealth of goods and services for society. It preserves a sustainable source of food, pathway for transportation, a venue for recreational and even household activities. It is also a source of irrigation and a venue for religious and cultural activities. Unfortunately, some of the country's major rivers, including the Bicol River, are undergoing various degradation. Among them are the result of urban growth and development, lack of concrete or sound land use policy, climate change, flood risk, agricultural activities, pollution, and increasing poor water quality. Several diversity indices are commonly applied to the study of fish composition and its distribution. Marine life is a bioindicator to assess the condition of the environment. Freshwater fish composition and distribution are primarily studied to assess the current status of rivers. Little is known on the current status and distribution of freshwater fish assemblages in Bicol River. That is why the study will characterize the freshwater fish assemblage within the selected areas of Bicol River. The morphometric characteristics, population distribution, and biodiversity of the freshwater fishes will be determined in the current study. The study will update the list of freshwater fishes in Bicol River and will provide essential baseline information for future fish survey and further conservation and management strategies for the studied area. The Bicol River is a medium-sized river in southern Luzon. It is situated in the Bicol Peninsula. The field inventory survey was conducted in the portions of the Bicol River along three municipalities of Camarines Sur, namely Bulac, upstream, Milaor, midstream, Canaman, downstream. In each site, two stations were selected, the urban and the rural area. In the municipality of Bula, the river in Barangay Panoypoyan and Barangay Pawili was selected. In the municipality of Milaor, the river located in Barangay Alimboyong and Barangay Amparado was selected. Lastly, in the municipality of Canacman, the river located in Barangay Ikin and Barangay Mayangawan was selected. Inventory of fish species was done by means